Welcome back to my channel Knowledge Chronicle.
Imagine trying to read a book. That's 3 billion letters long. Now imagine if that book holds the code to creating a human being, you. This isn't science fiction. It's the Human Genome Project, one of the most ambitious scientific missions in history. Today on Knowledge Chronicle, we're diving into how scientists decoded the human genetic blueprint, what they found, and how it's changing your health, future, and even the story of evolution itself. Stick around, because what you'll learn today will leave you in awe of the human body and the science behind it. What is the human genome? Let's start simple. Your body is made up of trillions of cells. Inside each cell is a nucleus, and inside that nucleus is DNA, your body's instruction manual. DNA looks like a twisted ladder called a double helix, and it's made of 4 chemical bases: Adenine A, Thymine T, Cytosine C, and Guanine G. The unique sequence of these 4 letters forms the genes that tell your cells what to do, like which color your eyes will be or how your body fights diseases. Now imagine this. All the DNA in just one human cell, if stretched out, would measure about 2 meters long. Yet it's packed tightly inside something invisible to the naked eye. So, what exactly is the genome? Your genome is the complete set of DNA, about 3 billion base pairs, that makes you. Why sequence the human genome? Scientists believed that if they could read this entire instruction manual, they could unlock the deepest secrets of human biology. Why some people get diseases and others don't. How to predict, prevent, and personalize treatment, and even understand our evolutionary past. But sequencing 3 billion base pairs? That was a mountain no one had ever climbed. This gave birth to a mega project in 1990, the Human Genome Project, or HGP. The mega mission. Let's put its scale into perspective. The human genome has approximately 3 billion base pairs. In the early days, it cost around $3 to sequence one base pair. That's about $9 billion to sequence the entire genome. Now imagine printing this in books. If one page holds 1,000 letters and one book has 1,000 pages, we'd need 3,300 such books just to hold the genome of one person. So yes, the HGP was massive, and that's why we call it a mega project. Who was behind the project? The Human Genome Project was a 13-year-long international collaboration. It was coordinated by the US Department of Energy and the National Institutes of Health, with major contributions from the Wellcome Trust in the UK and, countries like Japan, Germany, China, and France also played significant roles. The project officially began in 1990 and was completed in 2003, 2 years ahead of schedule. But what were the goals? Goals of the Human Genome Project. Identify all approximately 20,000 to 25,000 human genes. Determine the exact sequence of the 3 billion DNA letters. Store this massive data in digital databases. Improve tools to analyze this data. Share technology with industries for innovation. Address ethical, legal, and social issues, abbreviated as ELSI. Yes, decoding the human blueprint also raised deep questions like, who owns your genetic data? Can insurance companies misuse it? Can we ethically edit genes? So ethics became just as important as the science. How did scientists do it? 2 major strategies were used. First, expressed sequence tags, or ESTs. This focused only on genes that were active, that is, being expressed as RNA. Second, shotgun sequencing. This was a more comprehensive method. Scientists took the entire genome, broke it into random smaller fragments, cloned these fragments in hosts like bacteria or yeast, and used specialized vectors called BCs, bacterial artificial chromosomes, or YACs, yeast artificial chromosomes. These fragments were then sequenced using automated DNA sequencers based on the Sanger method, a technique developed by Frederick Sanger. Next, they had to align the overlapping fragments. Imagine solving a 3 billion piece jigsaw puzzle with no picture on the box. This required powerful bioinformatics tools, computer programs to analyze, store, and align the sequences correctly. Eventually, each sequence was mapped to one of the 24 human chromosomes, 22 autosomes, and the sex chromosomes X and Y. Fun fact, chromosome 1 was the last to be completed, in May 2006. What did we discover? Let's look at some jaw-dropping findings from the project. The human genome has 3.16 billion base pairs. The average gene length is around 3,000 bases. We have a total of about 30,000 genes, which is much lower than the earlier estimates of 80,000 to 140,000. 99.9% of DNA is the same in every human. Interestingly, only less than 2% of our DNA actually codes for proteins. Over 50% of discovered genes still have unknown functions. Repeated sequences make up a large part of the genome. Chromosome 1 has the most genes, 2,968, and the Y chromosome has the fewest, just 231. 1.4 million places in the genome have single-letter differences between individuals. These are called SNPs, single nucleotide polymorphisms, pronounced SNPs. SNPs are revolutionizing disease research, allowing scientists to trace genetic diseases and even human history. Applications and the future. This isn't just theory, it's changing lives. Personalized medicine. Based on your genetic profile, doctors can tailor treatments that are more effective and have fewer side effects. Early diagnosis. Genetic screening can now detect diseases like cancer, diabetes, or heart conditions, even before symptoms appear. Gene therapy. Fixing faulty genes is now a real possibility. Agriculture. Using genome data from plants like rice and Arabidopsis to create disease-resistant, high-yield crops. Environmental science. Using genetically engineered organisms to clean pollutants. And don't forget, model organisms like yeast fruit flies and nematodes were also sequenced to better understand human genes. Decoding the genome was just the beginning. The real challenge? Understanding what it all means. We now enter the era of functional genomics. How genes interact, how they get turned on or off, and how they form networks. It requires thousands of scientists from different fields. Biologists, chemists, computer scientists, data analysts, and ethicists. Biology is no longer about studying one gene. It's about exploring how thousands of genes work together like an orchestra to create life. So the Human Genome Project is not just a completed mission. It's a gateway to a healthier, smarter, and more genetically understood world. And if you found this journey as mind-blowing as we did, then don't forget, your one like, share, and subscribe will help us create more valuable content. Stay curious, stay informed. This is Knowledge Chronicle, signing off.